what I'd like to talk to you about is really the talk split in two parts. One is to discuss why we should actually measure platelet function, and then how we can measure that platelet function in a meaningful way in a clinical or pharmaceutical setting. So hopefully this is preaching to the converted, and you all know what platelets are, but just in case a few people don't, platelets are small blood cells, the smallest of the blood cells. You've got about 1 to 2 trillion of them in your body, and they live for about 10 days, which means that your body produces, give or take, 100 billion platelets each day. They float around your circulatory system, and because they're the smallest cell, they get pushed to the outsides of the blood vessels, so they're directly in contact, sensing changes within blood vessels as they float around the body. When they notice change, when they notice damage to the blood vessels, they become activated. As we can see here in the bottom picture, they change shape, they start binding together to form these platelet aggregates. And really, this provides us with an opportunity to use these cells, which are designed through evolution to sense changes in the body, as a biomarker. They're easily obtained from patients. They go to every vascular bed in the body, and using the changes that they sense during their 10-day lifespan can help us identify changes that are occurring within a patient. When I started working on platelets 20-something years ago, really all we discussed about platelets was them coming together, aggregating, forming clots, preventing bleeding, and in some cases forming inappropriate clots and causing thrombosis. But we now know there's an awful lot more that platelets are involved with, and platelets sense changes within the body. Platelets play a role within inflammation and sense changes with inflammation, atherosclerosis, angiogenesis and lymphogenesis. We're starting to learn that platelets also have a role within neurodegeneration, or at least sense the changes that occur within neurodegeneration. Platelets play a role in cancer proliferation, metastases, and they change in response to cancers within the body. And it's been well known that platelets play a role in infections. Infections such as dengue fever are well known to change platelet function, but a whole range of other bacterial and viral infections have been shown to change platelet functions, and platelets have a role in these infections. So I'm going to start talking you through very briefly about the role of platelets in thrombosis. And this is really the core business for a platelet. This is what platelets do the majority of the time. They float around the body individually. They sense changes, as I mentioned, and that can be changes due to damage of the vessel, or a device, a medical device that blood comes in contact with, changes in cancers and inflammations, as I've already mentioned. In response to those changes, they aggregate together. And in normal healthy situations, those platelet aggregates stop or cover the site of damage, stopping bleeding. However, if that platelet aggregation is inappropriate, it's too large, or it occurs at an inappropriate site, that leads to thrombosis, and heart attacks, strokes, pulmonary embolisms, etc. If, on the other hand, that platelet response is not strong enough, then we end up with bleeding. And that, we're not talking about just simply prolonged bleeding time when you cut your finger, for example, but we're talking about serious strokes or GI bleed. So it's a serious clinical bleeding situation. This is where I started my journey in platelet research. And this is work done by Paul Hayes as part of Alison Goodall and Ross Naylor's groups at the University of Leicester. And this study was performed just before I joined Alison's group. And it shows very clearly that platelet activity, here measured as the ability of platelets to aggregate together, that ability of platelets to become activated in response to ADP, which is a platelet agonist, was strongly correlated with a number of emboli that patients produced post-surgery. So in surgical situations, and this is surgery, carotid endarterectomy, where the carotid artery is opened up and the carotid a plaque in the carotid artery is removed. Following that surgery, obviously there is a highly thrombogenic area. There's damage to the vessel. Thrombus forms, but also emboli can be shed from that thrombotic area, float up into the brain, leading to strokes. What this study by Paul showed very clearly is that the higher a patient's platelet function, the more likely they were to have large numbers of emboli. What we did as a result of this was use an antiplatelet drug, clopidogrel, which is a drug that works to block the activation of platelets through ADP. And we gave a single dose of clopidogrel, so a single tablet, a 1 pound treatment on the night before surgery. And this was able to reduce platelet function by a really small amount, by about 5%. Before surgery, and about 10% reduction in platelet activation after surgery. This really small reduction in platelet function led to a huge reduction in emboli within the clopidogrel group and the numbers of strokes occurring within the group that took clopidogrel. So the take home from this is that really small changes in platelet function really can have a significant impact on real world clinical pathology. Of course, this isn't the only study that's shown that, and since that study was done nearly 20 years ago now, many other studies have shown the link between platelet function and clinical consequences through thrombosis or thromboembolism, or indeed bleeding. So that's great. We know that platelet function is important. We know that antiplatelet medication can be used to reduce platelet function, but there's an issue. And the issue is this, that not all patients benefit from antiplatelet medication. There are also serious side effects from antiplatelet medication, most notably bleeding, and those serious bleeds, the strokes, the GI bleeds. And patients' responsiveness to antiplatelet medication is highly variable. Because of that, around 6 years ago, a whole load of studies reported that showed very clearly that prescribing antiplatelet medication for primary prevention of cardiovascular disease really isn't effective. So even with patients with multiple risk factors, the evidence really doesn't support giving antiplatelet treatment for primary prevention. And the problem here may not be the antiplatelet drugs themselves, but the problem may be the platelet function isn't tested. So we have no stratification of patients based on their platelet function. Platelet inhibitors, antiplatelet medication, are not prescribed based on platelet function or even tailored based on platelet function. We have a one-size-fits-all strategy. And to show you why this seems to us like and to other groups like an area that we should be studying, it seems obvious that if you have risk factors for high blood pressure, you go and see your clinician, and they measure your blood pressure before deciding whether or not to put you on antihypertensive drugs. Similarly, if you have risk factors for high cholesterol, you have your cholesterol measured before deciding whether you should receive statin treatment or not. If you have risk factors that may lead to thrombosis, there is no test to measure platelet function. And this leads to a situation that was really clearly outlined in this review a couple of years ago, that states that platelet reactivity is a continuum within the population. There's a big variation of platelet reactivity within the population. Some people of the population will be at risk of thrombosis, some of the population will be at risk of bleeding, and there'll be a whole load of people who are at risk of neither and in the optimal zone. What we do at the moment in the traditional approach is by giving everybody the same treatment, a completely non-targeted platelet inhibition treatment. What that does is reduce down the platelet function of a few patients, but it also reduces the platelet function down of everybody else. Sorry, let me put that better. What this does is reduce down everybody's platelet function response. So a few people with high platelet function will move from having extremely high platelet function into the optimal zone of platelet function, but also a great number of people will move from the optimal zone of platelet function down into having low platelet function that puts them at risk of bleeding. Perhaps a better approach we could envisage is tailoring antiplatelet medication so that we can reduce down the risk of those patients with high platelet function, those patients at risk of thrombosis. And maybe we don't have one medication, we have a different strategy. So people who are not responsive to initial treatment get higher doses of medication. But also, and critically, we identify those patients who have low but normal platelet function and make sure we don't give them antiplatelet drugs, which pulls their platelet function into that zone that puts them at risk of bleeding. So as I said, that was a brief overview of how we think about platelets within thrombosis, but platelets play a role in a whole range of other diseases as well. This study came out a couple of years ago from the Framingham Heart Study, looking at nearly 2,000 participants and looking at risk factors associated with developing dementia. Clearly, age was the largest risk factor, but below that, we then have hypertension and then platelet function. We don't, at the moment, know why platelets have a role in the risk of developing dementia, or whether they're reporting on changes that relate to the risk of dementia. But clearly, the role of platelets within this disease is important. Platelets also change during aging. Platelet count has been known for a long time to change, to reduce during aging. So it stays relatively static during middle-aged, and then in late middle-aged and old age, we see a decline in platelet function in platelet count in humans, but we also see that in mice models as well. The data for the change in platelet function with aging is a little more confused. There have been a lot of studies over a number of years that show change within platelet function. Some of those studies show that you get an increased platelet function. Some of those show that you get a decrease in platelet function. Some of them show no change at all. What I think is happening, and there is a big caveat around this data because there is no consensus of this in the field. What I think is happening, and what our data suggests, is that you get an increase in platelet function during middle age, and then a decrease in older age in humans, and we can see that in mice as well. But there is a health warning to that, as I said. So that's just our current thinking on what's happening within platelet function. But there's a lot of discussion within the field about exactly whether this is genuine or not. We can certainly see changes in response to antiplatelet medication with aging. And you can see here, these are all patients who have received various doses of antiplatelet medication. And you can see after that antiplatelet medication, the older patients, no matter what the dose was, have higher levels of platelet function compared to the younger patients. There's also a change in platelets with disease. I mentioned earlier about the change in platelets with dengue fever. It's well known that in the hemorrhagic form of the disease, you get a reduction, a very drastic reduction in platelet count. And we'd like to know whether that is preceded by a change in platelet function. We don't know that yet, and we're trying to get grants to look at that, but that's something we'd like to see. We also know that there was a change in platelet activity during COVID patients. And we can see here that there's a change between, so the green dots here are the platelets and the blue dots are the monocytes. And you can see that in severe COVID-19, the platelets start to aggregate with the monocytes. And there's a change in platelet activation. And John Gibbons in Reading did some really nice work during COVID to show why this is occurring. So that was a very brief overview of why I think we should look at platelets. The take-homes are that platelets really sense changes in blood and blood vessels, and they do this in a whole range of conditions. They're incredibly easily obtained. We routinely take blood through venous blood through phlebotomy, but we're also working up our assays to work with finger prick blood with some pretty good success so far doing that. So they're easy attained, which makes them a really useful potential biomarker. And we think that they could be used firstly to identify the suitability of patients to antiplatelet medication and the efficacy of antiplatelet medication, as much as it's worth knowing whether people should go on antiplatelet medication. It's probably even more important to know whether that medication, how that patient is reacting to that medication. So is the antiplatelet medication that we're giving them having no effect at all? Is it having too much effect? And it seems quite astonishing, given how big these drugs are and have been huge sellers over the last 2 decades, that no patient who receives antiplatelet medication ever gets their platelet function tested. But also, I think, looking further ahead, we could use platelet function assays to measure disease progression and potentially age-related comorbidities. So that's why we should measure platelet function. I'm now going to switch to how we measure platelet function. And this really picture of John Gibbons here. John's my partner in crime for a lot of the work that I'm about to show you. And in fact, we're in the process of trying to set up a spin-out company from the University of Reading, a company called HaemAnalytica, which is really focused on taking the work that we've been doing and making commercial platelet function analysis for the pharmaceutical industry initially, but then also as diagnostic tools. So the first assay that I'll describe to you is the platelet phenomic analysis. And this is work primarily carried out in John's group on a BHF funded grant. And it's a platelet function assay that's designed to be high throughput, high dimensional, and work on flow cytometry. And it was published a year or so ago in Blood Advances. This work was primarily done. The assays were primarily set up by Nalin Creek in John's lab and Joe Dunster. The assay consists of taking a platelet flow cytometry assay, putting it on a plate, so that we freeze dry agonists in a range of concentrations onto 96-well plates that can then be stored for up to 3 months at room temperature. On the day that you get the patient in, open the plate, add blood, antibodies, either whole blood or washed platelets, whole blood or PRP, onto those plates, together with antibodies against fibrinogen and P-selectin. And we can then measure the concentration curve of response, the platelet response to a whole range of agonists. And Joe spent quite a lot of time automating this analysis system so that we can extract the data quickly and look at the platelet function from a whole cohort of individuals. Once we've got this information on the individuals, we can then use this data to stratify patients into phenotypic groups. So one of the major problems with platelet function tests that are available at the moment is that they tend to reduce platelet function down to very simplistic terms. So the point of care tests give a very narrow view of what platelets are doing. And platelets are, although they're small, they contain about 5,000 different proteins. They have a whole range of different responses to different agonists. And capturing that complexity, we think, is really important if we're going to understand how platelets are responding in different situations. So being able to take this complex data and then run that through machine learning algorithms to produce phenotypic groups with our patients or healthy donors is really important. Just to give you an idea of this, this was the process done on 150 individuals, and blue indicates low response to a range of platelet metrics, and red is high response. And we can see very clearly that we have these different phenotypic groups. We can have low responders at the bottom and high responders at the top, but also subjects with differential response, so low response to a certain platelet phenotype, certain platelet function endpoints, and high response to others. And we can link those phenotypes to certain clinical factors within the individuals as well. So here, for example, our low platelet responders seem to have high levels of, seem to be also the subjects with high BMI. So there is some link between platelet function and clinically relevant standard markers of health. Also, by taking the blood from these low responding donors and high responding donors, we can measure thrombus formation in these donors, and we can see that, as you would expect, people with high responding platelets produce larger thrombi than those people with low responding platelets. We're currently using this assay in a whole range of disease cohorts, focusing initially on ischemic heart disease, type 2 diabetes, but also looking at liver failure, frailty, infection, ITP. And we're starting to use this assay. We've used it in Reading, and it's been used in Oxford and Cambridge and Imperial. But it's starting to go into internationally. We have collaborations with Brazil and Chile and a group in New York. So if you're interested in using this assay in a different disease cohort, please get in contact with either me or John, because we'd like to expand our knowledge of how platelet function changes within different disease settings. To give you an idea of one of those studies that's been underway. So this is a pilot study we used in patients using the PPA assay. And this was conducted by Yasmin Mohammed, who's a PhD student with John and I, and Ilaria de Simone, who was a previous PhD student of ours. And using this assay, Yasmin and Ilaria measured platelet function in a whole range of healthy subjects and a whole range of patients. And then they added to the patients or subjects' blood 3 nanomolar of cangrelor. And cangrelor is an antiplatelet drug that inhibits platelet function, platelet response to ATP. And we can see that with cangrelor, sure enough, in both the healthy subjects and the patients, platelet function reduces. We can also see that there was actually no significant difference in the mean level of platelet function between the patients and the healthy controls at the start of the study. However, what can be seen is that there's a much bigger spread of platelet function in the patient groups. There's much more, there are many more patients with high platelet responsiveness and a few more with low platelet responsiveness. And what we did is we said, well, okay, let's assume that our healthy population have platelet function that is normal. If we take a cutoff of 2 standard deviations from the mean, above and below the mean, for our normal healthy population before cangrelor, and look what happens when we give the antiplatelet drugs, we can see very clearly that the whole population, a whole subject population moves down, and a few people in the healthy subject group have low platelet function that now falls below that 2 standard deviation threshold. So maybe at risk of bleeding. However, when we add cangrelor to the patient cohort, we can see that, yes, we do reduce down our high responders, and that's great. There's still a few people that have high platelet responsiveness, and maybe we'd like to add more in a clinical situation. We'd like to target these individuals to have higher levels of antiplatelet medication to reduce their platelet function further. But critically, at the low level, we can see we now have 36% of these subjects, of these patients, who fall below our 2 standard deviation threshold of the mean for our healthy population. And it's these guys, particularly the really low responders here, that we would be worried about in terms of bleeding risk. And that's exactly what was predicted in the review I talked through earlier. So I think we need to take this into this is a patient pilot study, and we obviously need more individuals, and we need to take it into bigger clinical trials. But it does indicate that perhaps we have a case for stratifying platelet function, stratifying platelet medication based on the platelet function of the individuals before and perhaps after they have received antiplatelet medication. So this is a different platelet function assay. The assay I've previously shown you is a static endpoint assay. We also wanted to look at the rate at which platelets become activated. Platelets, the natural environment for platelets is incredibly dynamic, blood flowing really quickly down blood vessels, and platelets have to react very fast in order to bind into a forming thrombus. And this work was, all the wet lab work was performed by Joe Mitchell, who's now in Birmingham. And again, the maths and analysis endpoint was done by Joe Dunster. And what we produced is a platelet kinetic assay. Again, this is a flow cytometry assay where we stimulate platelets at the same time as reading them, and we can see that we get a time-resolved change in platelet markers of platelet activation, in this case, fibrinogen binding, p-selectin expression, and an XM5 binding to negatively charged PS on the surface of platelets. If you look at the kinetics of platelet function, there's a large level of variation, which is what we want for a biomarker. It was also interesting to note that the rate of platelet activation is entirely independent from other markers of platelet function, such as the capacity at which platelets can respond to agonist stimulation. So it's something that needs to be measured separately on its own. Again, we measured platelet function in a whole range of individuals. We could see the different rate from different people. We then recalled subjects back and looked at their thrombus formation. And this is thrombus formation in collagen-coated capillaries. And we can see very clearly that there are more and larger thrombi formed in the blood of fast responders compared to the blood of slow responders. Interestingly, though, that result was shear dependent. So at low shears, so shear below 500 inverse seconds, there really wasn't a difference in response between fast and slow responders. However, at the higher shear rates, the 1,000 inverse seconds and 1,500 inverse seconds, so the shear rates that are more associated with arterial and perhaps pathological conditions, there was a significant difference between the size of thrombi formed by the fast and slow donors. As well as the size of thrombi changing, the structure of the thrombus changed as well. And what Joe did was, again, taking those collagen-coated capillaries, forming thrombi on them, she then flowed over those formed thrombi dextrans of different molecular weight that had different dyes on them. And using this assay, could then look at the density of the thrombi and where the dextran went into those thrombi or didn't. And you could see very clearly that the fast responders produced much denser thrombi than the slow responders. And again, this matters clinically because denser thrombi are likely to be more resistant to spontaneous breakdown or more resistant to lysis. So those are two assays that we do in the lab. They're quite complicated assays. They need the patient to be or the subject to be relatively close to the lab. So in Reading, we're lucky we have the Royal Berkshire Hospital down the road, so we can get samples and bring them to lab. Ideally, you want to analyse platelet function within an hour of phlebotomy, and that means you've got to have, you're constrained, tethered, both temporarily and geographically. There's a tether between the patients, subjects, and the lab that's going to do the analysis. We wanted to try and simplify things and break that up. And what we've come up with, and this is going to be the basis of one of the assays within HaemAnalytica, is a way of breaking that tether. So we still use the 96 well coated plates that have the agonist freeze-dried onto them. The blood is still added along with fluorescent markers of platelet function at the site of investigation and soon after the blood has been taken. But critically, we've been able to change this assay so that you can now freeze and ship those plates so that they can be bulk analysed by flow cytometry, and then using our bespoke analysis software and AI-based phenotyping can be done remotely. So what this does is really break that tether. No longer you can, with this assay, anybody can perform platelet function without any need for a specialist equipment or expertise. You simply need to be able to open the 96 well plate and be able to pipette, and without any need for specialist equipment other than to be able to freeze the plates before they're shipped and analysed by us. So we think this is will be a pretty big advance for running platelet function within clinical trials, but also potentially in the long run using platelet function as a diagnostic tool. We're also interested in simplifying this even more. The assay using within PPA is multi-dimensional. It's an incredibly useful assay. But can we simplify things even more for different situations? This led us to the idea of, can we develop a meaningful point of care platelet function test? And this is work that we've been following for the last sort of 4 years or so with Al Edwards, who was at the University of Reading, is now at University of Southampton, and Nuno Reis, who's in the University of Bath. And what Al and Nuno have done is develop work using a fluoropolymer MCF reel. So this is a melt extruded Teflon strips inside which there are 10 tiny capillaries. You can coat those capillaries, and then if you dip them into blood, you get capillary rise that can be measured on a standard smartphone. So I saw a presentation by Alan Nuno about 5 years ago and thought, wow, if this can work for platelets, it would be amazing. And sure enough, through 3 very talented PhD students, we've developed the test. And the test is extremely simple. The capillaries are coated with platelet agonists. You dip the capillaries into blood. The blood rises up and at some point reaches an equilibrium point. You can see that change in the equilibrium point, and these are pictures taken on a standard smartphone, and you can measure the height rise of platelet function. Decreasing rise is associated with platelet activation and increasing agonist concentration. So we initially started doing this with thrombin, and as you can see here, as you increase the thrombin concentration, we decrease capillary rise significantly. We can inhibit the action of thrombin using hiridin, and sure enough, the capillary rise changes back. Oh, sorry, capillary rise changes back to its normal level. And hiridin is blocking the thrombin's ability to cleave fibrin and activate platelet function. So it's blocking both the coagulation and the platelet activation within the blood. If we use a peptide called GPRP, and GPRP is a small peptide that binds to fibrin monomers but prevents them forming polymers. So Thrombin's still active within this assay, but no fibrin polymerization can occur. So the endpoint of coagulation can't occur. So what we've got here is platelet activation, but no coagulation. And yes, we still see a decrease in capillary rise. It's much smaller than we get when we have coagulation and platelet activation. As we'd expect, there's no gelation of the blood here, but we can still measure platelet activation by thrombin in these strips. We've developed this so it can work with ADP as well, and we can see that as we increase ADP concentrations, we get a reduction in capillary rise, and we can block that with cangrelor, or we can block that with EDTA. I should point out there's a health warning with these slides. The concentrations of ADP and thrombin that you'll see at the bottom are incredibly high. This is the concentration that is loaded into the strips. It isn't the concentration that the platelets see during the assay. What Rui has done is also take this assay and develop a rig so that we can automate the system. And this involves a light box and a Raspberry Pi and a servo motor that dips the strips automatically. And you can see here there's the blood, as the strips get dipped into blood, we get the capillary rise. And it can be done with either blood or plasma or PRP. And so what Rui's system allows us to do is measure 120 platelet function measurements, because each strip has 10 capillaries, or coagulation measurements within about 30 seconds. And this was published earlier last year. So the point of care test we think is affordable and accessible. The Teflon strips cost about £10 per metre. It's easily analysed either by Rui's rig or through pictures taken on a smartphone, and requires absolutely no operator expertise. It takes about 30 seconds and you can get a whole lot of information very quickly. Obviously, this isn't as dense information as the PPA test, but it may be useful in different situations, and particularly with this, we would be targeting primary care measurement of platelet function, and perhaps measurement of response to antiplatelet medication. So just to finish up with some final thoughts, I think actually cellular measurements really offer an exciting opportunity, diagnostic opportunity, and have huge untapped potential. I think platelets particularly, I mean, they are sentinels of vascular health. They go to every vascular bed in the body. They respond to a whole range of changes within the body. And they're easily accessed. So I think the assays that we've developed are relevant and accessible and relatively inexpensive. The future for this is really to try and enable patient stratification that will improve, firstly, the development of clinical trials. So one of the biggest problems with development of antiplatelet drugs at the moment is that we need huge clinical trials to do it. Actually, if we can stratify patients, we can reduce the size of the clinical trials. But also eventually improving the therapeutic efficacy, so making sure that we are not exposing people whose platelet function doesn't need to be inhibited to unneeded risk of bleeding complications. Potentially, this could be used, platelet function tests could be used as prognostic tests, particularly for disease progression, and maybe markers of age-related changes before those changes are symptomatic in other ways. As always, this is a huge team piece of work, particularly the Reading Platelet Group, both past members and present members, and our colleagues at the Royal Berkshire Hospital. And the funders, we've had we've had a lot of funding from a whole range of different organisations. If you want to get in contact, my email address is here, and we're always open for working with different patient cohorts and platelet function studies. Thank you very much.